Hi, good afternoon, good morning. This is our 24th Logical Reasoning in Human Genetics lecture. Today we are, I think, the easternmost, at least from Colombia, Tunis, among all our lectures. And today we have the pleasure of welcoming both Dr. Ainur Akilzhanova and Dr. Kushugulova Almagul, from both of them from the National Laboratory Astana at the Nazarbayev University in Kazakhstan. So as you have guessed, today we'll be talking about Kazakhstani genome, and the discussion will be about the challenges and application in clinical practice of Kazakhstani Reference Genome and Microbiome. And Dr. Joseph Lee from Columbia University will introduce the speakers, and then we can start. Thank you. Joe?
Hello, this is Joe, and I have the pleasure of introducing two of my colleagues. So we first met, Joe Terwilliger and I were teaching a human genetics a course that is exactly the same one that we're doing here in Astana, Kazakhstan in 2009. So that was about 15 years ago that we first met both Ainur and Almagul. And since then, that sort of kept the working relationship going for the past 15 years. And over the years, we have published several papers together, and then we're thinking about writing grants together and so forth. So to introduce Ainur first, she's a physician scientist, and she's the head of Laboratory of Genomics and Personalized Medicine at the Nazarbayev University, which is one of the leading universities in Kazakhstan. And her area of research of interest is cardiovascular disease and various other things, which she will talk about shortly in a few minutes. And the second guest who will be talking about her work is Almagul. We also met Almagul in 2009. And back then, they worked for a National Laboratory of Biotechnology, or some long formal name that I cannot remember anymore. But she is an expert in microbiome, and she's also has a lab there, microbiome lab. So that's when we met, and then, as usual, Joe Terwilliger does crazy things while he was in Astana, and then we can talk about that later. But without further ado, Ainur, would you like to start off on Kazakhstani projects that you are working on?
Yes, thank you very much, Joe, for the introduction. Thank you, everybody, for having us here and to giving us opportunity to talk about Kazakhstani research and what we've done for recent our days and years in our laboratory. Now, as you may know, Kazakhstan, localized in the Central Asia, is the largest country, 9th largest country in the world, and dominant Kazakh ethnic group. And this Kazakhstani lifestyle and population influenced by the nomadic lifestyle, long history of migration, and as a result, a mixture of Western Asian population, which formed our genetic background. And for us, and maybe for other researchers as well, it's crucial to understand genetic architecture of Kazakh population to proper investigate genetic basis of common diseases, common traits, and as well as inherited disorders. Here is shown that our territory is 2.7 million square meters, but population not so high, it's around 20 million population, and because of that, the density of persons per square kilometers is only 7.4. Here I showed some historical data and historical some information, but of course not to bother you with all of these words, but all of this historical data lead us to what we have now. I mean, what we have now, our diversity population in Kazakhstan. First of all, it's the Silk Road, We were part of this Great Silk Road, which goes through southern regions of Kazakhstan. Then in our history, we had a period where we belonged to Russia, like allegiance to Russian Empire, which is finished by this war in 1917. From 1919, we have occupied by Bolsheviks Red Army and became in 1920 Kyrgyz autonomous, in 1925 Kazakh Autonomic Republic. Then during Soviet time, government pursued to transform Kazakh nomadic lifestyle to settling of our population and colonized our region by Russian and other Ukrainian and other Slavic population. Then it was collectivization period, which is decreased Kazakh population for 1/5 of population was decreased. More than 1.5 million died during this time. Thousands of Kazakh population fled to China and survived, some of them, and it was several thousand people migrated to Uzbekistan, to Turkmenistan. Then also in Soviet time, it was in 1953, one big program led by Nikita Khrushchev. It's virgin and idle lands, opened vast grasslands of Kazakhstan, northern parts of Kazakhstan, to wheat farming by Slavic population. And as a result, many people from Ukraine, from Russia, from Belarus, from other parts of Soviet Union came to Kazakhstan, which has formed our genetic background. And of course, because of these things and other things, also we have some ecological problems, location in our territory, main space center. Then it also during 40 years, it was some nuclear weapon testing site in East Kazakhstan. This all lead to our some ecological problems and some problems in healthcare conditions. In 91, Kazakhstan declared sovereignty and independence. As a result, you can see that ethnic composition in Kazakhstan is around 70% is Kazakh population, 20% is Russian. Other population is less prevailed in our population structure, like Uzbek, Ukrainian, Uyghurs, Tatar, German people, and others. By religion, Muslim mostly Sunni, it's 70%, and Christians, mostly Orthodox Christianity, is at 26.2%, and we have other Christian, other like Catholic Christians and Buddha and etc., other religions as well. From historical point of view, it's interesting to say when we make introduction of each other to Kazakh population, we say, I am Ainur, my name is Akilzhanova, but except name and surname, we sometimes tell which tribe we are from. Concept of Jews and tribes reflects our historical organization of the nomadic society and in phases of kinship and unity. Each region in this Kazakhstan divided in juices. First, great juice is shown by red color. It covers territory of southern and southeast Kazakhstan. And middle juice, which is shown by yellow color, covers central part, northern part, and eastern Kazakhstan. And junior juice, or another name, small juice, consists of 3 tribes covering western Kazakhstan and some parts of Russia, with the former Orenburg Oblast. Each region occupied some geographical and climatic zone and determined nature of the economy and nature of the occupation, what these people would do in this juice. We have Kazakh folk wisdom, give the great juice stick to herd cattle, give the middle juice a feather to rule, and give the small juice spear and put it against the enemy. This folk wisdom showing what was mainly and what was main occupation in these 3 Jewish tribes. And this tribal division of Kazakhstani population, it's not, of course, exist now, but still we have like ancestors of that, not so many genetic diseases, inherited diseases among our population, because people know up to 7 generations their ancestors, and we are not have so much consanguinity in our marriages. Our national laboratory Astana belongs to Nazarbayev University, here shown as a picture. And Nazarbayev University localized in the Astana is the capital of our country. Here you can see on the picture Nazarbayev University, and this one is Expo 2017. It's a Nuralem. It's in the center of Astana. Our laboratory began to study genomic studies from 2009. And nowadays we have 2 centers inside of this big national laboratory. One of them Center for Life Sciences, where our laboratories exist, and Center for Energy and Material Sciences. Evolution of sequencing facilities and evolution of sequencing costs enable us to make genomic studies more and more widespread in Kazakhstan. Nowadays we have in our laboratory Novasic 6000 and Oxford Nanopore from long DNA fragment sequencing, which enables us to make not only reference genome sequencing, but also using this sequencing technology in frame of clinical applications. Cost per genome, if we will use Novasic 6000, decreases up to $18 per 1 gigabyte information. And if we will use Oxford Nanopore, it will be slightly higher, but still we can combine both technologies to make most comprehensive data of genomes. We have several scientific directions using in our genomic studies. First of all is the study of genetic architecture of population in Kazakhstan and Central Asia using high throughput platforms of NGS and TGS to create as a result a reference database of genomic variants. Secondly, we study molecular mechanisms of major multifactorial diseases in Kazakhstan, including cardiovascular, oncological, metabolic diseases, neurodegenerative diseases, to establish genetic epidemiology. Thirdly, we study tuberculosis and infectious diseases because tuberculosis is still high burden in Kazakhstan, despite decreasing in total number, but increasing in number of patients with antibacterial resistance. Fourth, we develop and implement new methods of sequencing, trying to implement in applied genetics in biomedicine, in populational genetics, agriculture, and sport genetics. And of course, when we found some novel mutations which need to be validated, we're now implementing functional genomics by using human-derived iPS-based cardiomyocyte modeling, CRISPR-Cas technologies, etc. Study design of our Kazakhstan Genome Project include, first of all, we would like and already recruited 500 Kazakhstani individuals from different regions of Kazakhstan and combine sequencing technologies. One of them is the short read sequencing using Novasic 6000, and the second one, long read DNA sequencing using Oxford Nanopore Promethean 48 machine. All data coming from these both technologies, from these 500 Kazakhstani individuals, form our Kazakhstani Reference Genome database, which will be used for evolutionary perspective analysis, for scientific fundamental research perspectives, and for clinical usage in our further clinical projects. First, our 500 samples and volunteers were recruited during our field works. We went to Torgay region. This is a central part of Kazakhstan. Also included all the people volunteered and included in this study from Astana, where many people from different regions of Kazakhstan gathered together. Then we went to Karaganda region. Here shown 2 villages, which is included Jansary and Timirtau. And last year we went to East Kazakhstan, where we recruited another more 200 population from Kazakhstan. Here shown some photos. We went during pandemic time. That's why we are covering all of these masks and lab coats and etc. Also, this our view was fear for our population, but still we were able to recruit our population to volunteer to our studies. Nowadays, of course, we increased our numbers in frame of human and other sources of genetic research. We included 400 whole genomes of cardiovascular patients during 2023 and now continuing this recruitment. 350 whole genomes we sequenced of the patients with post-COVID syndrome during pandemic time. 48 whole genomes of children with inherited disorders together with medical genetic laboratories in clinics. 24 whole genome of athletes of triathlon, these athletes who included in national Olympic teams. And also we continue targeted sequencing of athletes of Olympic teams in form of our grant projects. And of course, in cardiovascular projects, we included sudden cardiac death victims. Clinical data from all of this clinical part will form clinical database of genomic variants. And our 500 whole genomes of populational data will be as a control group as a Kazakhstani reference database genomic variants, and we compare between these both databases. Other sources and other objects is the bacteria, is microbiome projects. Then we study tuberculosis mycobacterium whole genomes. We study viruses during SARS-CoV pandemic time, and also some horses, because maybe you know Kazakhstan people eating a lot of horse meat. That's why we sequenced horses of Kazakhstani species. And maybe you know about antler deers, which is existing in East Kazakhstan in Altai Mountains. Also, we sequenced them to see what factors need to be maybe extracted for treatment and what mechanism of treatment of their blood. To make able to do whole genome sequencing and analysis, we need to create bioinformatic infrastructure, which was created during these 10-15 years. We established pipelines for analysis of NGS data and for analysis of third generation sequencing data. Here shown some pipelines which is established for NGS data and for Oxford Nanopore whole genome sequencing data. We made supercomputing clusters and created storage spaces for all of this data. This group led by our bioinformatician Ulugбек Kairov and our high performance bioinformatic cluster named Q-Symphony. Here are some characteristics of that, and of course, by every project, we're trying to increase our capacity. Some results. First, data of Kazakhstani genomes showing that we can sequence with high coverage, more than 30, which is standard. And here shows that standards is okay for us. Then total genetic variants identified in our Kazakh samples showed that some of them presented novel SNPs, which is not existed international databases. These novel variants might be a unique source to study our uniqueness and to study maybe some predisposition to another condition which is not exist in other populations. Distribution of novel structural variations to genome location. Most of them is prevalent in intergenic part, 50%, and intronic part is 40%. And other regions like some exons, some non-coding parts as well. And of course, we found some non-synonymous SNPs predicted by damaging by SIF, polyphen, and other in silico models, which is localized in different chromosomes, in different genes, and this only for few genome data. But while we're doing this 500 whole genome analysis, we will finding more and more SNPs, which is existed in normal population. Of course, studying our clinical genome data, we found many mutations related and associated with these conditions. Principal component analysis showed that Kazakh population clusterized in Central Asia together with other population groups. Here we analyzed different databases and datasets from HGDP, Horde genomic datasets, and also, which is interesting that this distribution and clusterization correspond with geographical map. And this is mostly Kazakhs localized in Central Asia. and still other parts of world maybe have also Kazakh population, which we need in future include in our study. Admixture plots of Kazakhs assuming different number of ancestral populations. These figures generated using data from more than 3,800 individuals collected in different studies like 1,000 genome, HGDP, Horde genomic projects, and admixture showing this different K5, K7, K10, which is showing number of ancestral populations. And you can see Khazars, Kazakhs, Uyghurs, and Uzbek, Kyrgyz. They are all belongs to Central Asian population and share genetic background, which is very heterogeneous comparing to other populations such as African or East Asian population, etc. Also, during analysis of only Y chromosome, we can see MDS plot based on pairwise genetic distance between Kazakh population and neighboring populations. Here in different, we divided them different groups, Kazakh all together, then West Kazakh population, North Kazakh population, Eastern Kazakh population. And here we can see that we are very close to Mongolian tribes, to Chinese tribes, to Uzbeks, and to Uyghurs. Russian and other populations are more far from Kazakh population. We used genomic studies and this genomic data to create panels and gene panels to study the genetic background of our patients. Here shown cardio panel development, including 96 genes, targeting exome sequencing using holoplex technology from Agilent. Here we included existed 34 and 21 panel genes, plus we included another genes which is not included in these panels, and make our own customized panel to study our arrhythmia patient and cardiomyopathy patients and other cardiovascular inherited disorders. Using this panel, here shown which genes included, we establish genetic diagnostics, and now working together with our clinician to establish clinical protocols to involve these genetic studies in our clinical practice. Here just shown as examples where we used in which projects. One of them is the primary electrical diseases, where we study using this panel potassium channel genes, and shown in different patients with AV blocks, with atrial fibrillation and ventricular fibrillation, different types of mutation which found in these groups. And by classification of ACMG, most of them pathogenic, and we found also non-pathogenic mutation. Some pathogenic mutation which exist in our population not exist in databases. That's why we need further validate by using human-derived, iPS-derived cardiomyocyte modeling. Another study, we used this panel in cohort of victims of sudden cardiac death who died outside of hospital, less than 45 years old, and they transferred to our forensic medicine center in Astana. During only 1 year, among 183 forensic cases, we found 37 victims died from unknown cardiac reasons. And we divided this patient these victims to non-structural changes of heart, and then 10 cases of them we diagnosed with cardiomyopathy post-mortem. It means that molecular autopsy can give us more data to establish more correct diagnosis. In this patient, as we show here, 63% mutations was found in the group of non-structural abnormalities. It means patient, these victims did not show any structural changes in their heart during autopsy. And 35 of mutation was found in group who diagnosed with cardiomyopathy postmortem. Most of them was diagnosed with myocardial infarction and was non-diagnosed before 40 years old. This panel can be used for molecular autopsy in forensic clinical practice. Here shown which genes mostly involved and in which genes mutation prevailed and what their pathogenicity according to ECMG. 3% of them is pathogenic and likely pathogenic. And of course, we found around 32% variants of unknown significance, which need to be validated further studies. We will validate by in silico models, by single sequencing, and etc. We used also our genomic data to establish minor allele frequency of variants, which is used for pharmacogenetic studies. One of the examples is using of genome-guided antithrombotic and anti-aggregant treatment in cardiac patient who were implanted left ventricular assist devices. In Kazakhstan is a large cardiological center, National Cardiac Center, localized in Astana, where from 2012 we perform heart transplantation and implantation of left ventricular and right ventricular devices. And in these patients is needed a long time, they maybe whole life they need to be under antithrombotic treatment and antiplatelet treatment. In this patient, we established also clinical protocols to use pharmacogenetic analysis and to provide some recommendation about monitoring of their treatment and about dosage of these drugs. In case of pathogenic variant establishment during genetic analysis, we can confirm diagnosis, we can guide treatment, we can monitor response, and provide testing for relatives and implement preventive measures. If we will find only benign variants, we follow standard medical care, and if will be a variant of unknown significance, in this case, of course, we follow standard medical care, and after update of information later we can recommend some changes in their management. As an example, here I am presenting some clinical cases. For example, a woman, 23 years old, she was admitted to cardiac surgery center. She had catecholinergic ventricular tachycardia at age of 13, and she diagnosed with idiopathic form of ventricular arrhythmia. Here shown her ECG, where you cannot find any normal QRST complex. And we sequenced this patient and found de novo mutation in ryanodine receptor 2 gene, which is localized in very hotspot area and shown as a pathogenic. We sequenced all relatives and parents of this patient and no found this mutation. But genetic counseling showing that her later children can inherit this mutation and lead further to arrhythmia. That's why she was treated with ICD implantation and with beta blockers, and this genetic study can help her management as well. Another patient, a male, 42 years old, Korean, he was diagnosed with idiopathic ventricular tachycardia. It's a paroxysmal form, and you can see here. And we sequenced him and his 3 sons and found mutation in exon 37 of ryanodine 2 gene. This mutation was inherited by his son, and for his son was recommended further monitoring of this condition by cardiologists to prevent arrhythmia in him. So we tried to implement the genetic testing and genomic studies to our clinical practice together with cardiologists, but nowadays we're also trying to include these genetic techniques in medical geneticists laboratories to our inherited conditions in the children, in oncology departments to study genomic profile of oncological diseases. Why is it changing? Okay, and trying to establish new types of data. Here is shown some photos, which is also presented by Joy Lee. You can see the black snow around our heavy industrial cities in Temirtau, in Karaganda, where localized our heavy metal industry. And you can see very polluted air in Almaty due to smoke and due to other reasons. All of these ecological factors lead us to do studies of influence of ecological factors, heavy metals, to memory decline, to dementia, to cardiovascular diseases. And we would like to create new project to apply for NIH grant, for other grants in Kazakhstan to study genomic data and to study some exposome in this patient to validate how these heavy metals might influence diseases in our population. Of course, I didn't present here all our projects, but still we have other topics as well to create some clinical trials to use our pharmacological combination of drugs against of oncological treatment, where in every case of patient we do genetic analysis of tumor profile, and also in cardiological patient we do also genomic studies, whole genomes of every patient with atherosclerosis, with family hypercholesterolemia, and trying to find what is clonal hematopoiesis risk to create as a factor risk to lead to atherosclerosis disease. And here in this photo shown our laboratory members working in a personalized lab, and shown all our collaborative partners. One of them is Columbia University. We're collaborating with them from 2009. Other parts joined during our research in different years. Also, we would like to acknowledge Laboratory of Bioinformatics and Systems Biology, led by Ulukbek Kairov and his team. And of course, one of the main challenges in all genetic studies to make genomic analysis from bioinformaticians. This is a bottleneck. That's why it's very important to us to increase members of this laboratory, to increase students and invite students to do analysis of genomic data. Some of our students, they entered to PhD programs abroad after finishing some analysis in our projects. And now they maybe in the future will come back. Thank you for your attention, and my colleague Almagul will continue. Then I will be happy to answer to all questions.
Okay. Okay, thank you very much. Hello, everyone. Just a minute, I just open my presentation. Okay, where is it? Is it okay?
Yes.
Yeah, okay, thank you. First of all, thank you very much for invitation and for the awesome introduction. And we go on from genome to microbiome. So the microbiome is one of many factors influences the development and progression of pathological processes. But the big but is that we can influence this factor. In the initial stage with the diet, later it can be some intervention with probiotic, antibiotic, or maybe some capsules with short-chain fatty acids. And sometimes we just can recommend to change the place of residence. Obviously, the main factor determining the microbiome is the human genome. Another important factor is the nutrition. And it is nutrition that determines the human health. We are what we eat, the famous phrase of Hippocrates. However, researchers have proven that environmental factors also significantly influence the human microbiome. The gut organ axis is a or multi-directional, multi-channel communication system that allow us the gut and the extra intestinal organs to communicate with one another. It's fundamental to understanding how the gut microbiome affects the human health. This figure from the publication from the article of joint research of scientists from France, Spain, Romania, India, and Pakistan. So, and as a consequence, microbiota dysbiosis can lead to dysregulation of bodily function and diseases, including cardiovascular disease, liver disease, cancer, respiratory disease, etc. And below you can see the link to this article, and this figure was designed by the researchers from Microbiome Research Center from Guangzhou, China. So the human microbiome reacts very quickly, and I think that it's an excellent tool for early diagnosis and prognosis the outcome of disease or outcome of the therapy. But first of all, we should understand that the most important is to understand what is a normal microbiome. It's a complicated question because the climate and nature that influence on microbiome, the climate change on our planet is diverse, and the food is diverse, and more diverse, but it's still possible. And microbiome is a tool that where we can take into account the age, we can take into account the norm by geographical location or some other factors. So Kazakhstan is really has a huge territory. And for to start this project to understand the normal microbiome for our population, we start and choose some territory with a duration about 2,000 kilometers and approximately the same latitude. So we created a team and went on expedition. We select some localities, both cities, big cities, small towns, and settlements, which are as far as possible from big cities and big roads, where the population lives mainly on natural farming. Thus, our first study included 502 urban and 149 rural participants. All participants underwent a comprehensive clinical and laboratory examination and sample were collection for microbiome analysis. All laboratory tests were provided for participants. Doctor on team provided free consultation because of in such remote settlements, it is inaccessible. In addition, all participants were given package of tea. Kazakh love tea very much. And according to our data, according to our questionnaire, in villages, Kazakh can drink up to 18 cups of tea. In 2011, enterotyping was carried first time by the team of Pierre Borg from European Molecular Biology Laboratory, Heidelberg, Germany, and directly done by Dr. Manimajan Ragumam. And using this protocol, we performed enterotyping and identified two enterotypes in Kazakh population, Bacteroides and Prevotella. And we found a predominance of Bacteroides enterotype in our population, regardless rural or urban microbiome. Only in two regions, Karaganda and east of Kazakhstan, we found the prevalence of Prevotella microbiome or Prevotella enterotype. The Bacteroides enterotype is associated with a diet high in animal derived proteins and fats. So according to our questionnaire in our study, we understand that this population prefer to have red meat. Additionally, this enterotype has been linked to disruption in causal integrity, including atrophic changes, epithelial cell loss, increased intestinal permeability, and is associated with the risk of IBD, colorectal cancer, metabolic disorders such as obesity or type 2 diabetes. In contrast, Prevotella enterotype is predominant in individuals consuming a high-fiber plant-based diet, rich in complex carbohydrates and polysaccharides, and associated with the risk of rheumatoid arthritis. According to our observation, according to our previous research, we understand that Prevotella in our population, Prevotella enterotype associated with spondyloarthritis, psoriasis, vitiligo, Hashimoto, and all of them is autoimmune disease. In this slide presents the features of the gut microbiome depending on the regions. In large cities like Astana and Karaganda, here marked as Akmola, but Akmola region, but the cities of this region, biggest cities is Astana, our capital. So in large cities such as Astana and Karaganda, the microbiome is characterized by the predominance of class 3D family and in the species level Bacteroides vulgatus, as well as a species Biophila wordsworthia. The last one, Biophila wordsworthia, associated with a high-fat diet and is associated with several metabolic diseases, including obesity and non-alcoholic fatty liver disease. Unfortunately, the incidence of this disease is, in our regions is increasing. A number of researchers have shown a close relationship between soil microbiome and human gut microbiome, which arose during evolution and continues to evolve. The dominance of bacteria such as Escherichia, Sutterella, Klebsiella, Desulfovibria, and Enterobacter is associated with the fertile Chernozem soils, with the high content of organic matter, high moisture, and this type of soil is typical for Astana region in our research. And another, for example, Turga region, characterized by kashtan soil, and I'm sorry, not kashtan, chestnut type, with a low humus content and used for pastures. Indeed, in Turga region, the population is mainly engaged in animal husbandry, and therefore the gut microbiome of this population enriched the bacteria such as Bifidobacterium, Ruminicoccus, and Biophila. And some of these bacteria the usual abundant of animal intestinal microbiome too. So we have found the significant differences in beta and beta and alpha diversity of between urban and rural population. Urban environments typically have distinct dietary patterns characterized by high consumption of processed foods, refined sugar, and fats, which may favor the growth of certain bacteria like Akkermansia, Dialister, and Bifidobacterium. Conversely, rural areas often have traditional diets rich in fiber, whole grains, and locally sourced produce, promoting the proliferation of bacteria such as Myxoacella, Ligulactobacillus, and Paraprevotella. And at the family level, distinct prevalence patterns for urban population exhibit Bifidobacteria and Desulfovibrio, and whereas the rural population, families such as Corybacteriaceae and Veillonacea. Corybacteriaceae and Veillonacea families belong to the Actinobacteria, but they have different nutritional requirements. In our study, the Corybacteriaceae is characterized in respondents diet by consuming fresh meals. And our early studies demonstrated that male milk consumption induces the growth in members of this family. Also, the gut microbiome of rural population characterized by the predominance of Veillonellacea family, which is known for its ability to metabolize lactate, a common compound found in fermented milk, and in our rural population, it's usual to use ayran. It's a fermented cow milk. A study of nutritional data revealed marked differences in eating habits between urban and rural population. On several parameters like salt, minerals, vitamins, proteins, and carbohydrate ratios. According to the WHO studies, Kazakhstan has a highest levels of salt consumption among other countries, and according to our result, we can see that the high level of salt consumption positively correlated with the urbanization index. So, for example, in Astana city is the highest, some rural population have lower. In the gut microbiome of the urban population, taxa with mechanism of replacing or altering the antibiotic action target are often identified, while with mechanism for inactivation in rural population. In rural population, our analysis revealed a predominance of genes associated with machinery toxin, mobile genetic elements linked to genus Klebsiella. The same gene family in urban population associated with Copracoccus. So, assessing the overall gut microbiome of Kazakh people and comparing it with our previous study, the data show a high prevalence of several genera, including Alistypis, Bacteroides, Bifidobacterium, Blautzea, Clostridium, Eubacterium, Fecalibacterium, Gemiger, and Prevotella with a prevalence of over 90%. This chart shows that the most genera are consistent with our previous data published in 2018. And with the data on multi-population data published in 2017 by Mancabelli. But some differences with previous, we think that it due to limitation of our previous research, because we focus mostly on the large city like Astana. But here we have some population from small cities and rural population. So here some photos. On the top of this page, we can see the photos on Turgai, and we can see that the well that used by population, and we can see the settlements are sparse, and there are only a few houses in the villages. The lower photo is the eastern Kazakhstan, Lake Zaysan, forest, and some settlements. And here my team, the team I consist from biotechnologists, biochemists, microbiologists, the specialist of machine learning, all of them young, and even some of graduated our Nazarbayev University. Thank you very much for attention. Question?
Oh.
I'm very quick.
Okay. Shall we start some discussion? Any questions for either Dr. Ainur Akilzhanova and Almagul? So if you have any questions, please go ahead. All right, so let me just start off. So Ainur showed that Kazakhs are a very heterogeneous group of people in the structural analysis. And in the structural analysis, you see several groups all coming together. And it makes sense because Kazakhstan is sitting in the Silk Road, and then you expect all to come together. Now, given that you have such a heterogeneous population, when you are looking to see how 90 some odd genetic markers for the clinical conditions that you were studying to see how well they predict their risk of diseases subsequently, have you taken into consideration level of admixture in those individuals?
Yeah. While studying genetic minimal allele frequency, we try to compare with our population data and see how it is prevail this allele in the same our population cohort. And this admixtures also influence in clinical part, because when we comparing with some Asians, for example, Chinese or Japanese cohorts, now comparing with European population, we have different data. That's why in clinical point of view, we need to compare with our populational genomic data as well.
So the reason I'm asking is this. When you have a genetic variant, let's just say for simple sake of argument, ApoE4, which is known to increase the risk of Alzheimer's disease by 3-fold, that tend to be consistent in white population and Asian population. However, when you look at the effect of ApoE4 in African American, you see that risk of ApoE4 associated dementia is close to 1 or 1.2, meaning it has a very little predictive value above what you expect to see in the general population for African American. So now if you're using those 98 genetic markers, SNPs, to help physician to treat or come up with some pharmacological agent to treat those persons who are at risk or having these conditions. How do you take into account that those of Asian background may behave one way, while those with Russian background may behave completely different, or other Turkish origin may have some other physiological responses that are quite different from one another? So when you are using those SNP markers to help the clinician, given that clinicians are not going to be aware of these sophisticated ideas of admixtures on the clinical outcome, how are you dealing with that possibility?
Yeah, when we include a patient or volunteer into study, we're asking about their ancestors up to at least third generation. If 3 generations they tell us that they are Kazakhs, we assume that mostly prevail Kazakh genes inside. If patients say that not involved other nations in his second and third generation, for example, only Russians, in this case, we mostly compare with European databases. But then altogether, we compare with all our genomic data, which exists from our 500 people. In our 500 people, mostly Kazakhs, 70%. Russian population, not so many. And that's why we need to go to other regions of Kazakhstan to increase numbers. No, at least like 5,000 whole genomes or something like that. For clinical purpose of view, we just using this up to 3 generation questionnaire and say what they have comparing to other existing data sets.
So are there data sets that you compare it to, say, from Russia, since there's obviously some historical similarity, relatedness? I don't know what's going on in Russia these days in terms of whole genome sequence, but is there much data that's out there or not?
No, most our data sets which we're comparing, it's from Southern Genome, then from HGDP, then from Asian populations, from like Genome Asia, 100K genomes, but Japanese genome. From Russian part, it's not so many. I think, I don't know exact data set which comes from Russia.
But do you have like ethnic Russians in your samples at all, given that that's a large part of the Kazakhstan population is ethnic Russian?
Yeah, yes, we have ethnic Russians up to 20%. In our genome data, we involved around 50 or 70 people, I mean, ethnically Russian. Not so many, but still we have it.
Yeah, because I remember when I was talking to some of the people in Kazakhstan, they all said that when a Russian and a Chinese have a kid, it comes out Kazakh. Is that true?
Yeah.
I also noticed that I noticed for Almagul, when you had your picture of Iran, that it said RussianFood.com. So maybe you better find a different picture.
It's not Russian, it's Kazakh food.
I
It's
know,
not
but on the picture,
in your picture,
the watermark
okay.
said RussianFood.com. So, you know.
Yeah, and so if I were to ask the same question to Almagul, when you have different admixture, do you see different microbiome distribution, given that having a certain different degree of admixture may represent different cultural and lifestyle practices, so that may also influence what you're seeing from your end?
Yeah, there are many publications that the influenced on some ethnic factor, but in our research, we have many. We have the same population. I know because we collect samples jointly, the same settlements. But we couldn't find any other clustering by other factors. So we find just nutrition, so diet. We find, of course, some pathologies because we collect the data on some comorbidities. We have differences, sex and ages. And what else? We have a very good clustering on longitude and latitude. That's all. Maybe because of it's not big population, because not big cohort. Sorry, but maybe later because now we are going to the west of Kazakhstan, and next year maybe south of Kazakhstan. Additionally, we invite some our colleagues and prepare joint project. And maybe we can invite the population from other corners of Kazakhstan. So maybe we find some ethnic, but now, unfortunately, no. Though we have Russian, yeah.
Isn't it almost measuring the same thing though? The ethnic variation with latitude, longitude, because of regionalization and the rural urban thing.
Yeah, it mostly depends on nutrition.
Yeah, which is correlated completely with,
yes.
Yeah. So they're all confounded. I mean, it's just hard to tease apart, I guess. Yeah. Makes sense.
Somebody else had a question.
Yeah.
So within a region, do you see difference between the older generation and the younger generation, where younger generation kids are eating westernized junk food, whereas the older person may stick to more traditional diet? I'm just guessing.
Yeah. That's why the difference is because small towns and fast settlements and big cities, For us, it was very interesting to recruit the respondents from far settlements, as far from the road, big road, because they have no any sugar drinks, they have no any burgers, they have no coffee, because they have a standard diet. And it's really interesting that in different villages, I mean, in different the villages, different regions, they have some traditions. For example, in East Kazakhstan, I was surprised that for breakfast they have soup. But for example, in central Kazakhstan, just usually in villages they have tea and have bread. In Zaysan, we try to go around this Zaysan, and we find some difference there because they have fish. They usually mostly use fish, but absolutely differently the small town near the Zaysan and villages. For example, maybe this some ethnic, because for example, in small towns, they live mostly Russian population. And they used some vegetables, mostly vegetables, fruits, but villages, they use mostly meat. Maybe this one.
So when you say the difference between the Eastern versus Middle, the distribution of different zoos, the Middle zoos and then great zoos and the small, they're differently distributed, right? So do you see a correlation among that and the region?
Let me answer for this. Yes, I think all of these people who already recruited in our study, mostly from Middle Jews, because the central part of Kazakhstan and East Kazakhstan belongs to Middle Jews.
Oh, I see.
And next our field works, we would like to go to West Kazakhstan. This is the small Jews. West Kazakhstan mostly deserts and near Caspian Sea, and of course food there is and diet is different, which also may influence our microbiome research. And also our plans to go to South Kazakhstan. South Kazakhstan is most densely populated, and we have a lot of admixture with Karakalpaks, with Uzbeks, with Uyghurs, and in South Kazakhstan, we can also see some maybe differences in microbiome, and also we expect some influence on genomic data as well. And from South Kazakhstan, we can go to Turkestan, to Shymkent, to Taras. It will be interesting data. So far, we have no chance to recruit people from that regions. And this 2-3 years, we plan to go there.
And interesting that the average age of South Kazakhstan is higher in comparing other part of Kazakhstan. And the density of centenaries in South Kazakhstan higher.
That's interesting, because I would have thought young people might move to like the cities, it's like to Almaty or something for work or for school. So it's
but Almaty, the population of 25% of centenaries in Kazakhstan lives in Almaty. Only 6% lives in Astana cities. I don't know the villages.
That makes sense, I guess. Yeah, because Astana is such a young city. Also, why would they be there? Yeah. I mean, that's like a migration history question almost. I mean, but that also is something that surprises me, because you said that sampling in Astana would mostly be from that, ethnically from the Middle Jews. But isn't everybody in Astana from somewhere else? Because like nobody was really there like 30 years ago, right? Hardly anybody was there, right?
So in Astana, we could find people from every part of Kazakhstan. In south, from West Kazakhstan, from East Kazakhstan, and I have no data now to tell what prevail. I mean, Eastern Kazakhstani population or the Northern Kazakhstani in Astana, but still here all people from whole Kazakhstan presented. And of course, when we study Astana population, we trying to establish what origin. From which city or which village he came to Astana.
Yes,
but
I mean,
diet almost will be same.
right, but that's why it's actually interesting, because you can have the genetic variation in Astana without the microbiome variation, and you get the microbiome variation from sampling outside, so that way you can control for the differences, right?
Yeah.
Because
And now,
otherwise it'd be correlated, the genetics and the microbiome would be correlated if you sampled villages, but in Astana they'd be different, or probably Almaty also.
Now we have plans to study microbiome and genome together to see correlation, as you told. Yeah.
But I mean, it's also interesting to look at the sampling from the cities, because there you wouldn't expect them to be correlated. So you could look at the correlation by looking at the villages where the genome and the microbiome would be different. But in the cities, you'd expect the genome to be different, but not so much the microbiome. So then if you're looking at health effects of both, that would be more informative, right? To have the heterogeneous samples. That could be really interesting.
Yeah.
Oh.
It's very good
Joe,
suggestion.
I'm not, I get your idea, but the microbiome is somewhat stable. It changes, right? But just because somebody moves to the city, in this case to Astana, pretty recently, doesn't mean that, you know, in Astana you have a pretty homogeneous microbiome. So there's all kinds of studies now that look at developing countries where people live in very rural areas where they live traditional lifestyle, and you see that the microbiome, essentially from birth, how it's acquired thereafter, is very different from what it is in the West. And in the West, we have a hard time finding similar microbiome compositions, even in people that we might view live in a rural area, some farmers somewhere, so then they start, at least in the US, they look at, you know, more traditional people, the Mennonites, the Amish, people that use raw milk that hasn't been pasteurized and all this type of stuff. So there they find microbiomes that are a little bit more similar than you might find in developing countries. But even those people seem to be kind of westernized already. So I don't know, you live in a beautiful and highly varied country, landscape-wise, lifestyle-wise, and nature-wise. So I think looking at the microbiome is a wonderful way to inform yourself about some of the changes that are taking place in your country, whether or not that is clinically relevant or it's more a descriptive observation. You can see that in the West, our lifestyle certainly hasn't been all good for us when we think of diabetes, obesity, heart disease, and you name it. So I'm not sure that research can prevent those changes, but it's certainly a good way to document some of those changes. So one question I had for you on the genetics, I mean, with all these different ethnic groups, I mean, we talked mostly about looking at the genetics, at what makes these different groups maybe different and whatnot, but you presumably also have very unique, family patterns with lots of kids. Do you have inbreeding in some parts? I mean, do you have unique genetic diseases that you find in Kazakhstan or maybe just the region thereof, that you're aware of, that they are very rare elsewhere? You know, those are all things that maybe you could take advantage as a geneticist to, kind of take advantage of what you have there in your country, as opposed to kind of what we do in the US or Europe or the West in general.
Yeah, as I told this, our historical division for 3 Jews, it means in every Jews it was several tribes. And before, historically, these tribes had nomadic style, and they are going from one village to another village along this territory, and not allowed to marry inside of tribe. And usually we ask up to 7 generations to know that we are not relatives, and then they can marry, a boy and girl from different tribes, from East Kazakhstan to West Kazakhstan.
7?
No, up to 7 generations. Yes.
For example, male line though, only the male line, right?
Yes, male from male part. But usually they said, okay, you are my relative because your grandfather have a brother from my grandfather. It means we are not possible to be married in future. That's why we think that this might lead to less amount of inherited disorders. We're thinking like that. But of course, we are not telling that nothing exists from inherited disorders in Kazakhstan. Before we had large families, like up to 10 kids in one family, 10 children. But nowadays, 3, 4, this is average. Even 2, 3, it's also average in average family in Kazakhstan, especially in younger families. That's why to tell that many inherited disorders or rare disorders, not so. But we cannot say that they excluded.
Yeah, it's like
This is
there's a lot
interesting
of variation, sort of, because so many people have gone through the region over time, and they're
Yes.
avoiding marrying close kin. So it's actually probably a good thing, right? It leads to all this excess variation.
But also, other part might influence that international marriage. For example, we have many Uzbeks, many Russians, many Koreans, and Kazakh can marry Russian, or Russian can marry Korean, and etc. This is also interesting to see how this might be influencing our genomic data and how to interpret in frame of clinical point of view.
Do you have good relationship between all these countries that are in your area that all end in stans? You know, there's Tajikistan, Uzbekistan, Kazakhstan, you name it, more. With all their different ethnic diversity within these countries, and sometimes they're distributed one group among different countries. So it might be a good way of kind of looking at the region holistically from a genetic standpoint, if you could collaborate with some of these other countries.
Yeah, recently we had the Illumina Days in Nazarbayev University, and people from Uzbekistan, Tajikistan, then Armenia came. And of course, if would be possible, we would like to establish like Central Asian cohort for genomic whole genome data. And it will be good, maybe source of data for all of us to study, because in Kazakhstan, maybe in some parts in far village, we can find some homogeneous genetic group, but in Tajikistan or maybe other countries, maybe it will be possible to find some tribe or something which will be homogeneous, but still heterogeneity among all Asian population, Central Asian population will be very high. But we would like to establish Central Asian cohort to study, and we talked about collaboration with them. And moreover, we would like to enter together with our collaborator from South Korea, Dr. Johnson So, to go to Central Asian genome, not Central Asian genome,
Right.
Genome
Wow.
Asia 100K, if will be possible. We talked about that before pandemic, but after this COVID-19, we just paused.
Yes, like everybody else, all our plans went up in smoke. Thank you very much.
You know him, Harold, right? You know So Jung-sun, who.
Yeah, I do. Unfortunately, I have to go. I'm sorry about that. I got another call here that I have to take. So thank you very much, and I'll just hang up and you keep on with your discussion. Nice meeting you.
Yeah, I would guess, like, in Harold's question about the ethnic diversity, you're right. I mean, because most of Central Asia is kind of flat, so people move freely. But like the Pamirs and stuff in Tajikistan, there probably are some isolated groups that are much more unusual because of the geographic inability to get anywhere to find mates. So that obviously could be interesting.
Yeah, it's possible.
So, question for Almagul. I assume when you're doing microbiome, you're using stool samples, right?
Yeah.
How correlated is a stool sample as opposed to saliva samples if you were to do microbiome?
Yeah, we have several projects. We try to correlate it. Unfortunately, maybe because of limitation of small sample size, we couldn't find any strict correlation.
But different.
We have on rheumatoid arthritis, centenarians, and now we start to collect with cardiovascular disease, and this population, this cohort will be much bigger. If rheumatoid arthritis about hundreds and psoriasis about 100, but this we will have, we would like to have about 600 cohort. Maybe we will find something. Unfortunately, now, no.
Almagul, you made also microbiome of Alzheimer's disease patient. Is it any correlation among them between stool and saliva microbiome? No?
No, no. We try to we moved from 16S to shotgun, and now try to analyze all our samples using shotgun, because it's not a simple prognostic. We can find them metabolic, maybe through metabolic ways, maybe through the analyzing immunome. Maybe we can find something, because the mucosa immune should be synergetic at any organs. Maybe through this we can find some correlation, but taxonomic, no, it's absolutely different.
I guess it makes sense, right? Mouth.
Yeah.
Different bacteria, yes.
Yeah. I would guess.
But at least in my experience, people have a quite significant resistance to providing stool samples as opposed to a saliva sample, so that when you have 30% participation, then you have no data. So that's, I don't know what the participation rate is in your study when you ask for stool samples, but it's probably, I don't think it would be that easy.
It would probably be, it also might be, what I was just thinking is I remembered the time I spent with the Kazakhs in China. I know a lot of them are now in Kazakhstan because of the problems in China. I mean, it might be interesting to look at their microbiome, given that the fermented milk stuff that they were eating is much rawer than the stuff I've had in Kazakhstan, because it was much more primitive. So I bet there's a lot of differences there, and it could be quite interesting, because I know the people that were my guides in Xinjiang when I was there in 2009, 2010, they're all now in Almaty because they lost everything in China. So there might be some interesting samples there.
Like
it might be interesting to compare these groups.
Like of Ainur, PCOA analysis that there was for like a geography map. The same we can do by a microbiome. It's absolutely, we find that China microbiome, Kazakh, and even Kazakh, inside some regions, and USA absolutely differently.
Sure. I was thinking about the Kazakhs in China, because they're such a their culture is so much more like old, more traditional in a way. It'd probably be quite different microbiome just because of the different way that they ferment the milk. Could be interesting.
And also, when you look at people who are in Astana, you may want to ask when they migrate to Astana, so that if they migrate from rural area 30 years ago, that would be different than those who migrated 10 years ago and so forth. So because the lifestyle probably would change for those who migrate 30 years ago as opposed to 5 years ago. So you may want to sort of like, when did you move in? And that may give you some insight into how rapid the changes in the milieu of enteric conditions. So that's something to take into consideration, perhaps.
The interesting fact is, for example, in our cohort, that some people live for many years in urban, in Astana, for example, but in microbiome we find some taxons that are associated with living in villages before, in детстве, как
Yeah.
in childhood.
Yeah, in childhood, yes. So it was for us, it was interesting. We were surprised, and the interesting that the health status of these people is higher than compared to other population. It's interesting because usually we need just few months to change microbiome for and said that it's mostly urban than rural, maybe 12 months. But here is 10th, 20, 30 months. Yes, that's why it's also interesting.
Is any of that related to perhaps the probability of using antibiotics and stuff for disease where rural people might not have it as much as in the cities? Because I know here, at least, we over-medicate everybody in the cities with penicillin and stuff.
Yes, it's, we think that it's very important to collect the data on some medication that they used during their life. Unfortunately, we have no.
I was just wondering, because I guess that's
nutrition, yeah, diet, and some laboratory, clinical laboratory data.
I was just wondering, I was assuming that might be different between the rural people who move to the city versus people who've always been in the city in terms of how often they go to the doctor and get drugs versus just deal with it. Because my parents, they never bothered getting antibiotics. They were country folk. And they're just like, we don't need the doctor. But in the city, they go every time they get the sniffles. It's interesting. Anyway, I think we probably have to start wrapping up because it's almost 11:30. I just realized that we've been going for almost 90 minutes. But I really want to thank you both for coming and presenting your work. I think it's been great. Are Yusuf and Mariam here?
Yes.
Okay, yeah. Because I know you guys need to use the thing at 11:30 for something else. So, are there any last questions or comments or anything either one of you want to say? Other than we want to thank you greatly for coming here and presenting. It's, I think, a good exposure for people in North Africa since they're also trying to start this. And you guys started this like 15 years ago with genetics, and you built something quite impressive in Kazakhstan compared to when Joe and I were there for the first time. You know, it's been very amazing. And I think it's really nice to compare what's going on in your experience with the North Africans. I hope that everybody can learn something from each other. So we really appreciate it.
Thank you very much for invitation.
Yeah, for invitation and for this opportunity and for fantastic discussion. Thank you very much for some new ideas.
Yeah, and hopefully we'll have more over time. We'll get to interact more.
We would be glad to see you in Astana. Not only you both, Joyce, but also our North African, maybe future collaborators, and we would be happy to share what we will have, and maybe we can learn something from our colleagues from North Africa. We would be happy to see in Astana. Hopefully we will organize some conference and we will invite definitely both of you. And maybe you will join to our this field works in North Kazakhstan or maybe West Kazakhstan or South Kazakhstan. We will see. We will inform it.
And you have a lot of experience doing that sort of field work in all these different locations. And I mean, our North African colleagues are thinking about doing similar things. So it's a good, you can help.
Okay.
Okay.
Joe?
Yeah, I think it was a fantastic discussion of multi-omic, if you will, of a different population than North Africa or European American cohorts. So it was a very nice discussion of how things go when you're trying to start from the beginning to create a reference population, to expanding it to by microbiome and try to integrate that with genomics as well. So I think this is a very nice introduction, and there's something to be had from both North African colleague as well as us, and we had a nice lively exchange. So hopefully Next time, colleagues from North Africa will present something similar along that line, and then we can all learn from something there. So, well, thank you for joining us, and see you next month.
Okay. Thank
Okay.
you.
Thank you.
Thank you. Bye.
Bye-bye.
Thank you so much. Bye-bye.